It is remarkable how 20 years have changed concept of plant signaling, from a situation in which even the word signal and transduction received no mention, to special issues devoted to the critical topic. Signal transduction is used by plants to coordinate their development and to sense and respond to fluctuations in their surroundings. A substantive number of genes will be involved with the signaling necessary to specify cell position and fate in developing leaves or roots. A plethora of signaling molecules for cell development must be present with their transduction machinery because cells continually change position during growth and must continually update their spatial and temporal conditions. Although sugars and auxins may be simple molecules, they have long been seen as signals in vascular tissue differentiation. Furthermore, other complex signal transduction circuitry must be present to ensure the stability of morphogenesis, the overall form of leaves, roots, and flowers. In chaotic environmental conditions, although some plant characteristics are resistant to environmental variation, many others respond plastically, adding an extra layer of morphogenetic and biochemical complexity that is largely unique to plants. Cytokines, opsicins, ethylene, and gibberellins, as well as more recently discovered barcinosteroids, jasmonates, and peptide hormones, are agents that help to strengthen plasticity. As the environmental context changes, so will the strength of connections vary between the proteins and other molecules that make up the network. A strong spatial dimension underlies signal transduction. For example, plants must be able to detect gradients in signals, such as light, and resources, such as nitrate and water. Plant development itself also is decidedly polar. The spatial dimension is satisfied in many ways. Cells place receptors, channels, G proteins, and kinases in specific membranes. Some signaling protein complexes are permanent, such as the relatively stable and perhaps hardwired constitutive photomorphogenesis signal SOM-COP9. Other signaling protein complexes are likely to be ephemeral and formed immediately as a result of signaling. There are at least 600 receptor kinases in Arabidopsis, and most of them are membrane-bound. Incompatibility and disease defense signal transduction use receptor kinases. After ligand binding and autophosphorylation, such kinases may act as nucleation sites for the construction of ephemeral signaling complexes that contain many proteins. Many other GTPases, 14-3 proteins, kinases, and phosphatases may cluster around these nucleation sites, forming kinase cascades, and the regulation of specific transcription factors, for instance biphosphorylation, will complete the pathway. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon, AJ.